Great. All right, folks, please welcome Corey for introducing her research.
Thank you all for coming on the last morning here. Thank you to the organizers for keeping everything moving smoothly. I'm hailing you from the University of Buffalo, and I'm going to present today on a project we've had going in my lab for 2 to 3 years now. We're sort of in the middle of the transition as we sort of phase out the first cohort of students and into the next. So that's why you're getting me instead of the people who've done most of this work today. But we started this project in the Western New York region around Buffalo, in part because we were coming at these questions from the idea of we want to learn something about the fundamentals of invasion ecology. And for better or worse, the Great Lakes is a really wonderful place to study such things because there's lots of that going on. So one of the sort of ideas, like foundational ecological ideas that provides the framework for the kinds of things that we're excited about is how the sort of environmental context of any ecosystem is providing the framework for the particular species that we find there. So what are their life histories, what are they eating, what does the food web structure look like, and how does that get disrupted when there are stressors in that ecosystem. So again, toward a nod to our Great Lakes origins here in Western New York, there's a lot of this kind of work going on in the Great Lakes themselves. So this is kind of a classic figure from the GLEAN project, where they developed this idea of accumulative stress, which is basically all the ecosystem stressors rolled into one, right? So this is pollution, eutrophication, and the intensity of invasive species. And if you sort of boil that all down into one metric, you can look at the places that are more or less impacted by these multiple stressors on our aquatic ecosystems. And so from the aquatic ecology side of things, we were interested in looking at how these things correspond to what we're seeing in the context of invasion ecology. So if we think about some of those foundational concepts about like how habitat quality can change and influence community assembly, how does disturbance regime influence the frequency that these kinds of species turn over and their resiliency, can we look at all of that specifically with a lens for invasion? So in thinking about this in Western New York, one of the sort of ongoing species invasions is from the round goby, and I probably don't need to provide a ton of background information on the round goby, but one of the reasons why we chose round goby is because although it's now established in all of our Great Lakes, there are still active invasion fronts going up into the tributaries of the Great Lakes. So in terms of the time frame of catching this invasion and learning something about how invasion progresses and how the impacts of invasion develop over time, this is sort of a good system, making the best of a bad situation. Because they've been here for a little over 30 years in North America, there are places where we have really well established communities with round goby as a key part of that process. So you might have heard something about this with thiaminase this week, for example. All the lake trout folks love them some round goby. But in the lodic systems, we're seeing a little bit of a different story because the lake trout are not so much a player in those particular food webs. So looking at the impact of round goby as they move into lodic habitats from the Great Lakes is sort of an interesting question for learning about the basics of invasion and simultaneously tracking the impact of this invasion as they move into inland waters. So we've been doing a bunch of surveys for round goby across Western New York. We go out and we do fish surveys, so we have a full assessment of the species that we find there, all of the fish species. We've also, in addition to this, been taking samples of all of the primary producers, both in the stream and in the riparian area, as well as assessments of the stream macroinvertebrates, including some mussel surveys. We have been pairing that with water and habitat quality assessments. So we basically go out and throw the kitchen sink at these problems every single place that we go. And so we have this nice, really robust data set for as much as we can sort of understanding all of the things going on in each of our sampling sites. So here's a map of what's going on. We've hit 21 sites so far in total in the state of New York. This is a little complicated, so I'll sort of walk you through it. We're here in the Western New York end of the state in the Buffalo metro area, for those of you who are familiar. You can see the sort of tributaries are largely flowing into Lake Erie. This is the sort of eastern end of Lake Erie here, and then this is the Niagara River here on the left, separating the United States and Canada, just to help orient you. So we've got a number of tributaries that are running into Erie and Niagara through the Buffalo metro area. So one thing you might notice is that these we've got a lot of urban systems, and if you go sort of out toward the skirts of where these tributaries are starting, there's a lot of agriculture in the area as well, for those of you who've been there. Each of our sample sites is noted here by either a square or a circle, depending on when we first started hitting that site and doing our surveys, and then the color corresponds to the proportional abundance of round goby in the fish assemblage at that site. So there are a handful of places here where there are goby absent, where goby have not actually become established yet, and then there's sort of a range of densities of round goby in the other sites going from this sort of orange to red color. There's kind of 5 separate tributaries that we're looking at here. One of them sort of branches off, so we're considering it a little bit separately. But one of the things that you'll notice is that we sort of don't have equal representation of round goby across these systems. So we've got one that's completely round goby absent at this moment in time, and others where they're really, really abundant through the vast majority of the stream length. A lot of this has to do with one of the things we've considered in this sort of assessment of invasion is that there are impoundments in a number of these places, which is probably not terribly surprising given that we're looking at urban and agricultural streams. But interestingly, we have sort of a smattering of things going on here. So for example, Ellicott Creek, which is sort of up in the Amherst suburb area. For those of you familiar with the area, there's a natural waterfall there. So there's some bedrock happening that's precluding round goby upstream invasion into some of these places. Whereas somewhere like Smoke Creek down here in sort of the southern end of our sampling area, there's these very, very slight changes in the bedrock elevation that I would wager that if there's a big flow event or something that they would likely be able to pass it eventually, but for the time being it's keeping them out. In other places there are anthropogenic impoundments. So in the Buffalo Creek here, there's a series of low-head dams that are keeping them out. So that's probably going to last for a little while, as long as somebody doesn't put them in their bait bucket and drag them around. Whereas up here in Tonawanda Creek, we have what I'm calling a temporary barrier, where it's basically this big nasty log jam that probably will get washed out at some point, but we're sort of seeing it being a physical barrier to upstream movement at this point in time. But the thing that I guess I shouldn't say I'm excited about, but one of the things that we found this last year of sampling is in Murder Creek, a very ominous location for such things. We think we identified the actual invasion front here. So this is a fairly long stream length up Tonawanda Creek, and then it branches toward the south to Murder Creek. And in a very frustrating sampling day, we had a really, really high abundance at our sort of second site here. And then when we went to our upstream site, we were there for a good hour and a half, saw absolutely no round goby. And then, of course, like the last say in the hall, we get one tiny little round goby. It was extremely frustrating, but we think we may have identified the actual invasion front there. So all of this to say that we have sort of a really good smattering of places where round goby have been established for a really, really long time, but are precluded in their upstream invasion. And we have examples of where the active upstream invasion is ongoing. So in terms of asking questions about how does invasion biology work, we've got a really good sample of stream sites going that we're going to continue to sort of poke at over the course of the next few years. In addition to that, we also know that there's sort of things going on outside the Buffalo metro area, and you probably heard a number of talks about Lake Champlain this week. And one of the things about Lake Champlain is that folks are pretty concerned about round goby making it in. They're sort of coming at it from both ends of the system, and there's concern about what might happen to Lake Champlain if round goby were to get in. So knock on wood, that's never going to happen, but just in case, it doesn't hurt to have some before data, which is a very lovely and rare thing in ecology, as we're often reacting to these things after the fact. So we're trying to basically take what we're learning in Western New York and apply it to the situation going on in Lake Champlain so that we can sort of better inform management or mitigation strategies in the event that round goby get there. And there's a whole bunch of folks working in the Lake Champlain Basin, so many thanks to all of the work that we're able to build off over there. But one of the things that we're interested in is not just looking at the lake itself, but looking at the tributaries into the lake. So there's a number of folks doing these same kinds of things in the lake itself. But last year we were also able to go out and sample some of the New York tributaries to Lake Champlain to sort of get this before data. And we basically do the whole thing, the fish survey, the macroinvertebrates, we take tissue samples from all of these things, bring them home, and we'll be able to sort of do the same kinds of analyses to see where we might find sites that are more or less vulnerable should round goby eventually show up. So we have 4 sites in Lake Champlain along these watersheds here, and the plan will be to go back this spring to do some more tributaries on the Vermont side. So the sort of like overall specific questions that we're getting at with this particular study is to what extent this environmental context that we're seeing in these buffalo streams, the Western New York streams, how does that influence invasion success and influence the impact that round goby are having on these systems? And can we use that information to better prepare ourselves for future invasions? So one of the ways that I'm excited about looking at this is through the context of a food web lens. So we know that round goby have been integrated pretty well into a lot of the lake food webs, but how does this look in a much smaller habitat where we're looking at generally smaller bodied fishes? And we've seen in other systems, particularly from some work done in Michigan, that they come in and sort of get aggressive with other benthic fishes. So there's a competition happening there both for habitat and for food resources. So one of the ways we wanted to tackle this was by doing a stable isotope analysis to reconstruct what food webs look like in invaded and uninvaded reaches of these streams. So just to give you a brief primer, make sure we're all on the same page, we're basically using carbon and nitrogen isotopes to sort of reconstruct what that food web looks like with all of the samples we've taken from the variety of surveys that we do. And if we think about how this might look in terms of competition between the invader and native species, if we sort of look at all of those stable isotope values for one species compared to the other, we can talk about like what niche breadth looks like, how much overlap there is between those niche spaces on our bivariate plot here. And then if we want to know the difference between round goby and a benthic species like some of our native darters, we can sort of look at that in isotope space to sort of ask these key questions. So first we'll go over some of the diet data that we had. Here we're looking at the actual contents of the gut. So we have the samples from our invertebrate communities along the x-axis and the gut contents of the round goby themselves here on the y. This line is the one-to-one line. So if they were eating everything in the proportion that it exists in the ecosystem, everything would be along this line. So what we're seeing here is they're sort of avoiding a lot of things here. There's a lot of invertebrate species that exist in these systems that they're not really eating much of, and there's sort of a handful that they're sort of targeting. They tend to be some of the more abundant ones like midge larvae, some of the caddisfly species that are common in the species, and a handful of mollusk species. If we then look at the same sort of data but from our stable isotope data, we put it through some mixing models, and then we get the proportional diet composition from all of those same invertebrate species here. So they're here and colored on the left. Along the y-axis here we have two different sites from our Ellicott Creek samples. So just as an example, two sites from Ellicott Creek, 1 and 2, and we've got two different bars on each side. The left one is a rainbow darter, and the right one with the asterisk on it is round goby. And so the thing that I sort of take away from this is that Ellicott 1, which is our downstream most site where round goby have been established for the longest period of time, there's a really high diversity of diet items comparatively, both in our rainbow darter and our round goby. But in the upstream sites, the rainbow darter is eating far fewer things. The round goby diet composition is relatively similar. So there's a difference between sort of what's going on at the invasion front and what's going on at those sites that have been established for a long time. So as round goby become more abundant, as that invasion becomes established, darters are sort of partitioning what they're eating rather than focusing on particular things that maybe are the preferred items. They're sort of diversifying their diet and eating lots of different things. If we then sort of move to this question like, okay, so darters might be shifting what they're eating in response to round goby abundance, then what does that actual question of niche overlap look like? Can we actually identify evidence for competition? So here we're looking at the sites relative to their distance from the mouth. So again, the lakes being the source of this invasion. So moving upstream are newly established sites. And here we're looking at the proportion of the niche overlap. So how do those two circles overlap in isotopic space between round goby and darters? So we see that there tends to be an increase in the overlap of those things as we move upstream to relatively newer invaded sites. So when round goby first arrive, there's a big overlap. And as we saw in the diet composition, darters are then sort of partitioning their diet to eat other things over time. And if we look at how that corresponds to round goby proportional abundance, so sites that have been invaded longer with larger abundance, they tend to have sort of this decline in the overlap of the niche space tends to decline as round goby become more abundant. So we're seeing this increase in the niche partitioning as round goby become more abundant and more established. And if we look at sort of just the nitrogen values of this that indicate the trophic position, so where are we in sort of that food web level? Are we eating at the top of the food chain or the bottom? We see that as we move upstream, both round goby and Johnny Darter and darters tend to have higher trophic positions, so higher nitrogen signatures. But if we look at how that changes relative to goby proportional abundance, we see that there's sort of distance between the darters and the goby sort of increases as that round goby proportional abundance increases as they become more established in these sites. So we're seeing this sort of evidence of competition where as they become more established, darters are sort of shifting what they're doing in terms of their food base to account for the round goby competition. So in terms of the implications of these sort of preliminary findings for what's going on in Lake Champlain and potentially other future sites of invasion, we see the risk for invasion sort of across a variety of habitat types, but we do see the greatest niche overlap and the greatest evidence of competition happening at the invasion front. And as they become more established, darters are sort of shifting to potentially less desirable food items to sort of avoid that competition. So the overlap in these diet items could be a problem for places, particularly on the Vermont side of Lake Champlain, where there are a number of listed darter species that are of concern. So as round goby move into these habitats, if they do arrive in Lake Champlain, this will definitely be something to keep an eye on, particularly in sort of a rapid response fashion. So the future directions for this, we're going to be going out and doing sampling on the Vermont side of Lake Champlain, hopefully. And I'd like to do a greater integration of the assessment of riparian areas, because my sort of big ecological idea that I'd like to work toward is sort of mapping an overlap of invasion in aquatic spaces with what's happening particularly in the Buffalo area, with invasion of riparian areas. So if you've ever been out to the Buffalo River, for example, the riparian area in a lot of places is essentially a monoculture of Japanese knotweed that has been demonstrated to disrupt sediment and carbon dynamics between aquatic and terrestrial ecosystems. And so I'd like to know what sort of impact this has on invasion, if it's adding to that idea of cumulative stress in these systems that the darters and native species then have to deal with on top of this invasion happening. So we're going to go out and assess some of these things this summer in a little bit more detail. We'll have sort of this full accounting of what's going on through as much of the ecosystem and all those dynamics as we can. And I'd like to sort of work toward this idea about whether or not invasion in riparian areas is going to facilitate invasion in aquatic spaces in this idea that I'm calling cross-ecosystem invasional meltdown, where, similar to Dreissenids facilitating the invasion of round goby in the Great Lakes, does a terrestrial invasion near an aquatic space with high interactions have an impact on the invasion progression in the aquatic invader as well? So we'll be working on that over the next few years, but I should wrap up by thanking all the number of students and collaborators who've helped us with this, especially the New York Water Resources Institute for helping us get this off the ground. And I think that's about my time, so I'll take questions if there's time, but thank you for coming.